Thank you very much for the introduction, and thank you very much to everyone for turning up on a Monday evening to come and hear me talk about pain. Why does it exist? How does it work? And hopefully I'll give some evidence towards the end as how we might be able to more effectively treat it. So this is a drawing taken from 1793, where unfortunately we didn't have particularly good anaesthesia or analgesics available, your best bet being some rather large people who might hold you down while you scream. If we zoom in on this little plaque in the top right-hand corner, we can also see what members of the public thought about doctors at the time. So you have people named things such as Christopher Cutgut, Peter Putrid, and Abraham Abscess, and so on and so forth. So hopefully our faith in medicine has improved over time. So in this evening's talk, I'll try to cover, first of all, what exactly is pain, to make sure everyone's on the same page. Talk a little bit about why it exists, because I think some people always think, well, it's a really unpleasant, bad thing. Wouldn't life without it simply be a lot better and a lot easier? And then what we also need to think about is the problem of chronic pain. So there is a difference between the ouch side of life and then actually where pain can have a real impact on how people go about their everyday life. And then sort of in the last half or so, I'll give you some examples, some research going on in the lab for some of the things we are trying to do to look at how we might be able to more effectively treat pain. But first of all, obviously this is the A.V. Hill lecture, so I thought I should at least give some link between me and A.V. Hill, which is dubious, but I can manage it a little bit. So he won the Nobel Prize for his discovery relating to the production of heat in the muscle. And I'm very much a neuroscientist, not a muscle scientist. But he did work a lot on how pH modulates muscle function and hemoglobin, that of course being the way by which oxygen is carried around the body. So here we have some papers where he's looked at the influence of the external medium of the internal pH of muscle, and here he's looking at the combination of hemoglobin with oxygen and carbon monoxide and the effects of acid. And this is how we can link to the research in our lab and most of my research career, which is that we focus a lot on pH, how low levels of pH acid can cause pain. That's what I did my PhD in, and that's what lots of work still ongoing in our lab is focusing on. And also we work on this rather what I would call magical beast, the naked mole rat. And we'll come on to this at the very end of the lecture. But the one thing to say about the mole rat at this point is it can survive at very low levels of oxygen. And so I think if A.V. Hill had the opportunity to study its hemoglobin, he would have been quite impressed, because it's an animal that maintains a very high affinity for oxygen all the way through its whole life. Whereas for most of us, when we spring out of the womb, our hemoglobin changes its type as we don't need the high affinity to bind as much oxygen. So what then is pain? Well, if we go back in time, so we're here in the 1600s, Bruegel the Elder and Peter Paul Rubens made some paintings of the 5 senses. So here we have the sensation of sight, so this lady is surrounded by lots of different things that she can be looking at. Sense of hearing, so now she's playing a musical instrument, there's a bawdy crowd in the background singing, and there's lots of different musical instruments. The third sense is that of taste, so now she's being presented with a table laden full of lots of things to eat. Then we move on to the sense of smell. So now she's outside and she's surrounded by lots of different things that she can smell, lots of flowers and so forth. And then lastly, of course, the sense of touch. So here she is cradling an infant and surrounded by lots of things that she can physically interact with. So we have the 5 classical senses: sight, hearing, taste, smell, and touch. But if we look more closely, we can perhaps have an insight into some other senses that we've learned more about over time. So if we focus here on the sense of smell, the lady is outside, naked, and that is important. So we can interpret that for 2 reasons. It may say something about how artists of the time wanted to portray women, or it could say something about biology, which is that it's her private garden, it's a warm afternoon, she doesn't need to wear clothes. And the reason she knows it's warm is because of thermoception. We have nerves that enable to tell us if it's warm, if it's cold. So I'm going with the idea that actually her thermosensation is working well, it's warm, she's in a private garden, and hence she's not wearing any clothes. If we go up here for hearing, and we look more closely, we can see that she's looking back at us while she's still playing a musical instrument. And this is what we term proprioception. You know where bits of your body are in relation to each other without having to constantly look and move things around based on where you can see where they are. So you know where your body is in time and space without having to physically think about it all the time. So these are all very important, but I would argue the most important thing is actually highlighted down here in touch. So if we zoom in there, we can see that although she's cradling an infant, right next to her on the table are lots of medical instruments that we would be expecting to be used in the 1600s. And although these appear sharp, they're certainly not the sorts of things that I would want to have to be impaled on my body if I was undergoing surgery. So although we've got the 5 classical senses, pain has very much always been there throughout history. So what exactly is it? Well, I'm a member of the International Association for the Study of Pain, which is a mixture of clinicians, physios, nurses, scientists, anyone who's really interested in pain. And there's a really exciting committee, the taxonomy committee, where they get to decide what words mean. And this is their definition of pain. An unpleasant sensory and emotional experience associated with, or resembling that associated with, actual or potential tissue damage, which is a very long-winded explanation. So there's a few key things to think about there. So in terms of this resembling that associated with actual tissue damage, so if I pick up a baby and I hold it at head height and drop it on the floor, what will happen? It'll cry. Hopefully it'll cry. So is the baby in pain? It's crying. We think so. So, yeah, resembling that associated with actual tissue damage. But you don't have to go back that far into the medical literature to see that we didn't think that neonates could actually feel pain. Surgery was conducted without analgesia. So although it may seem obvious to us that a crying baby is in pain, and therefore we should treat the baby as though it is in pain, that is not, or has not always been the case. So what about this sensory and emotional experience? Well, that's where we need to contrast pain with another word, and that word is nociception, the neural process of encoding noxious stimuli. So neural, we're switching a nerve on, noxious, something unpleasant. So if I get up in the morning, I make a cup of coffee, I pick it up, it's too hot, I will go ouch, and I'll put it down again. This is nociception. My nerve got switched on by the heat of the coffee mug, and my nerve has done its job. I don't walk away from that mug being depressed and anxious about approaching a coffee mug for the rest of my life. It's a one-off sensory experience. By contrast, for somebody living with chronic pain, there'll be that sensory part of things, but there is also the emotional component. If you're living, for example, with osteoarthritis, sitting still may be causing you pain. That will impact your ability to engage with normal everyday activities. Therefore, you get less enjoyment out of life as you're less able to do things. And we find in a chronic pain population that levels of anxiety and depression are much higher than they are in a non-chronic pain population. So just as a get out for some potential questions later on, I'm officially a professor of nociception, so I study how nerves get switched on, whereas I don't really do anything to do with emotions. So pain is complicated, and we make it a bit easier to study by just avoiding the brain and the emotions altogether. So that's obviously incorrect. We should study things holistically, but you can only do so much yourself. So we focus on the sensory aspects of pain. So what exactly does that look like? Well, if you open a textbook, pain will look like this. We've got an end organ, in this case it's the skin, and we've got these grey things which are some hairs sticking out of it. And very broadly, we have two sets of sensory nerve fibres. We've got some shown in blue, which are mechanoreceptors. They're involved in the sensation of touch. And we've got some shown in red, nociceptors, those nerves that have evolved to detect purely noxious stimuli. Now, there's lots of different subtypes and differences between the two, but one key property that distinguishes a mechanoreceptor from a nociceptor is how they respond to an ever-present stimulus. So because we're now in November, I'm assuming most of you are wearing socks. But you probably need to wiggle your toes to remind yourself you're wearing socks. And that's because your mechanoreceptors don't constantly need to tell your brain you're wearing socks, you're wearing socks, you're wearing socks. But if you stimulate them, you'll be reminded that, yes, you are wearing socks. By contrast, the nociceptors do not really adapt, which is really important. Again, back to my coffee mug example, if I pick it up and go, ouch, that's hot, oh, no, it's not really, and then pour it down my throat, there'll be a lot of damage happening. So nociceptors tend to keep firing that signal until you remove the stimulus. Their job is to detect something that could cause damage. So regardless whether it's a nociceptor or a mechanoreceptor that gets activated, an electrical signal flies off the spinal cord, and this is a single cell. So from the little toe to the bottom of your spinal cord, those are some of the biggest cells in the body. The way that the nerve works at its ending, how it can detect a hot stimulus and acidic stimulus, is because that proteins are made within the cell body, which is located in something called the dorsal root ganglia. And they get trafficked down to the end of the nerve, and that enables that nerve ending to detect whatever's going on in the environment. And studying what happens at a nerve ending is very, very difficult because the nerve is embedded in lots of other tissues. And so what we do, and many others in the world do, is we study the properties of cell bodies because we can isolate very simply the DRG from rodents and model species that we regularly use, and mice and naked mole rats, rather than trying to study what's going on at the actual nerve ending. So like every model, there are limitations, but you can only go so far with each individual technique. So once we activate the nerve, the signal goes off the spinal cord, and occasionally a nerve will synapse, connect with one other nerve and go straight up to the brain. About 5% of nociceptors do that. The vast majority go into a complex circuitry of interneurons where that signal is processed. And that's something we don't really understand too much about, but we are beginning to understand ever more. And that will eventually lead, hopefully, to a motor output, so you respond to the noxious stimulus. So this is the textbook example. If you were a mouse, it would look a bit like this. So the brain is off the side here, and there's the spinal cord of the animal going down towards the tail. And you can see that on both sides of the spinal cord, there are these white balls. These are the DRG, just like in this diagram here. And your body is organized in segments. And that's what we can see at the bottom of the screen. We've got some skin from the back of this animal, and these white fibers here represent the axon bundles, which will communicate stimulation of the skin to the DRG, to the spinal cord, and then eventually up towards the brain. So that's the anatomy of how it works, but why does it exist? We've been talking about this unpleasant sensory and emotional experience. We can look for two different ways as to why pain might be a good idea. So we can look throughout the animal kingdom, and we can see evidence for nociception, ouch, and nociceptors, nerves that detect ouch, throughout the animal kingdom. So we've got sponges over here, right the way through to Homo sapiens, us over here. And all these species shown in grey are where we've either identified nociceptors or nocifensive behaviour. So for example, here we have C. elegans, a 1 millimetre long worm. Some of these worms are males, but they don't really need those. The majority are hermaphrodites, and they have 302 nerves. About 24 of those function as nociceptors. So even what you could argue is a basic organism with a rather low-level nervous system has dedicated a huge chunk of it towards going ouch. Which kind of makes sense, because if you're wriggling through the soil and you come across something that's dangerous, you need to respond to that, move out the way, and that will enable you to live and fight and reproduce another day. The medicinal leech, shown down here, is a segmented animal. It moves in concertina fashion, and each of those segments contains a ganglion, just like our dorsal root ganglion. When you look inside there, you can see end cells, nociceptor cells. And again, the leech will respond to unpleasant things and respond accordingly. Fish is where life gets a bit complicated. They definitely have nociceptors, and they definitely respond to damaging stimuli by showing specific behaviours. But do fish have emotions? So humans, we definitely have emotions, and we'd probably argue that most things own mammals over here definitely do. Now, I've got 3 cats. I can't really see what they're thinking, but I can imagine, and one of them gets bullied a bit by the others. They're probably a bit more depressed than the others. They're definitely more anxious than the other two. But what about fish? Well, if we look at the bits of the human brain, or the mammalian brain, that we know are involved in emotional processing, and if you stick a human in an MRI scanner and you, under consenting conditions, hurt them, and you look at which bits of their brain light up, some of those are involved in emotional processing. And fish share some, but not all, of those parts of the brain involved in emotional processing, which you can also activate by giving a fish a stimulus that causes that fish to go ouch. So do fish have emotions? Do they not have emotions? I work with mammals, so I don't need to answer that question, but in the scientific field, it's a real contentious issue as to whether they're just going ouch or whether they have potential for anxiety and depression, which of course can have a major impact on how we deal with fish farming and everything else to do with fish. But fundamentally, what Darwin says down here is correct. Any variation, however slight, if it be in any degree profitable to an individual of any species, will tend to the preservation of that individual. A nociceptive system enables you to detect what's going on in your environment and respond accordingly without getting damaged. And I just want to give you one example of this, of an organism that most of us would probably take for granted. Thankfully, it's November now, which, although it gets colder, does mean there's less chance of fruit flies appearing in your kitchen. Now, fruit flies are the bits that we find a bit annoying, but before the flying stage is a larval stage. So what we have here is a fruit fly larvae, and you can see in yellow the nerve fibres. So what these scientists have done is they have made those nerve fibres that we know are involved in nociception, they've made them express a protein that we can switch on with blue light, and we activate those nerves. So let's see what happens. The larvae is wiggling along, the blue light comes on, and it does this coiling behaviour. It starts rolling all over the place. Blue light goes off, and eventually it will come back to crawling normally again. So again, the blue light is on, the animal starts rolling. So you could say this is a bit unfortunate. Why are these horrible people stimulating these nociceptors and causing this animal to do the rolling? Or you can ask the question, why does the animal do this when you stimulate the nociceptors? So what you're going to see in this video is a wasp attacking the fruit fly larvae. So some wasps do this by putting an ovipositor, an egg-producing little appendage, into the larvae where it will give its eggs into that larvae. So let's see what happens. So here we've got our wasp, and the larvae are a bit difficult to see. They're these things with the black eyes wiggling around, but the video we pause at the crucial moment. So the wasp's looking around, it's stuck its thing into this larvae here, and then watch what happens. So it's trying to attach onto the larvae in order to impregnate it with all the eggs. And that larvae is wriggling and rolling and flips the wasp onto its back and rolls off. So this is why this behavior that you saw over here, this wiggling behavior, has evolved. Because when a wasp tries to impregnate the larvae, it activates this rolling response, and that poor wasp got flipped off, and we didn't have an impregnated fruit fly. So there's a reason why nocioffensive behaviors have evolved. It's not there for fun. But we can also see the importance of why we need the ability to detect noxious stimuli when we look at humans when things don't quite go as planned from a genetic point of view. So this boy here has a condition called congenital insensitivity to pain. So congenital, it's a genetic condition he's been born with. Insensitivity to pain, can't feel painful things. If we look closely, we see you've got no digits on either hand. He's got an ulcerated left knee. He's having to stand by leaning on this ladder. And if you ask him to open his mouth and stick his tongue out, he can't. He doesn't have one anymore. So what's happened? Well, if I go back to my coffee mug example, I would pick it up, go, ouch, it's too hot, and that's the end of the event. By contrast, this person would pick it up. They wouldn't know it's painfully hot. They might then proceed to pour boiling hot liquid down their throat. Now, obviously, just because they can't feel it doesn't mean it won't do any damage. And unfortunately, quite often what happens is these people have injuries. Injury leads to infection. Infection leads to amputation. If you took away any of your pain sensation, as an adult, you know how to interact with the world to limit damage. But obviously, with very young children, this is quite complicated. So there are different forms of this condition, different severities, and there is a family in the UK where the parents are both healthy, but all three children, now in their 30s, have this condition. They're all fine. Got numerous knocks and bumps, but not to the extent of this poor guy here. So why does this come about? Well, there are different reasons. Some mutations in genes affect the anatomy of the nervous system. So I'll just give you two examples here. If you have a mutation in these genes, you don't have the nociceptors there. So anatomically, you cannot detect ouch because the nerves required for that are just no longer there. Other people appear to have perfectly normal anatomical nervous systems, but they have abnormalities in how that sensory sensation can be activated and sent through the nervous system. And so one of these genes is something called SCN9A, and this encodes what we call a voltage-gated sodium channel, which the non-physiologists in the room you'll have never come across. But if you've ever been to a dentist and you've had something unpleasant done, like having a root canal or something ripped out, hopefully the dentist first would have given you an injection of something. And what he would have injected you with is lidocaine, a non-selective inhibitor of voltage-gated sodium channels. So you couldn't feel anything that was going on while it was unpleasant. And SCN9A is one of those voltage-gated sodium channels that's particularly important for going ouch. So if that gene isn't working properly, you have a sensory abnormality, and in some people that can lead to a lack of pain. But as we'll see later on, this gene is particularly interesting because it can do all sorts of different things with regard to pain. But we'll come back to that later. So hopefully I've convinced you that though pain might be unpleasant, it actually is a good thing to have. But it's also not a plastic sensation. So under normal conditions, we've got pain intensity on the y-axis and stimulus intensity on the x-axis. Again, you're all wearing clothes. They're producing a mechanical stimulus, but that mechanical stimulus does not make you go ouch. So not all stimuli cause pain. But at a certain point in time, the stimulus becomes great enough and you start to scream. And that's good, because that will mean you'll avoid the bad thing and hopefully look after yourself. But the system doesn't stay like that. If we have an injury, everything shifts to the left. We increase the sensitivity in the system. Clinically, we can use two words to describe this. Allodynia is where stimuli that used to cause no pain now do, and hyperalgesia is where stimuli that used to cause some pain now cause a lot of pain. And so, for example, if you've ever been sunburnt on your neck, what one day was a nice warm shower, the next day becomes burningly painful, you need to turn it down. So again, this is a good thing. I go running quite a bit. If I fall over and hurt my knee, the knee will be injured. It will be more painful. Therefore, I'm likely to look after that injured part of the body until it recovers. And what happens usually is processes cause the blue curve to become the red curve, and depending on the extent of the injury, you come back to the blue curve within a bit of time. And of course, if you listen to your body, you'll probably encourage that healing process rather than keep trying to do things when you are in a state of pain. The problem is that for some people living in chronic pain conditions, they've got stuck at the red line for no real reason. And so a lot of work we do in the lab is trying to understand what causes the blue curve to become the red curve, and can we make it go back again? So can we identify new drug targets? So I'm sure many people in the audience have taken ibuprofen, diclofenac, these sorts of drugs. and most of those are functioning by preventing the production of one substance, or one group of substances, that causes this blue curve to become red. So when you take that tablet of ibuprofen, your pain gets less, you're switching off what we call this sensitization process. Now again, you might think, well, why would we want pain to get even worse? Surely even if you're injured, it would be better just to, for the pain to go away. But you can do some quite nice studies across the animal kingdom to demonstrate that, again, this gain in pain is actually a really important thing. And so some collaborators of mine several years ago published this very cool paper looking at squid. So what they did is they let these poor little squid get eaten by this rather big fish, but under different conditions. So some of those squid were uninjured. Other squid had an arm amputated. And other squid had the arm amputated under anesthesia. And the idea is that when you do that injury without anesthesia, you'll develop this sensitization. And in a sensitized state, just like me with my running, where I'll go running less to let that injury recover, the animal's behavior will change, which could be beneficial for its survival. And that turns out to be true, as we can see in these data here. So look at the percentage survival. So this is an uninjured squid. Most of them survive, 3/4. If we just anesthetize a squid, it doesn't really have any effect on what happens. If we injure a squid, more of them do get eaten, but it's a lot more that survive than in this final group here, which is where the squid was injured in the presence of anesthesia. So this sensitizing process to increase the gain in pain doesn't happen. And that influences the animal's behavior. So normally the squid will just be swimming around. Once it has this injury, it becomes hypervigilant. It will respond more rapidly to a fish coming towards it. Whereas this poor one that's been amputated under anesthesia, it's got the inability to swim as well, but hasn't got this gain in pain signal. Its behavior has not changed. So you can do similar studies in the wild or under laboratory conditions as shown here. So the study shown here is for diagrammatic purposes. People don't expose mice in labs to foxes, but what you can do is expose them to fox urine. Mice will usually run very quickly if they're exposed to fox urine. And what you can do is in models of chronic pain, a much further away stimulus of fox urine can cause the same response. So the mouse becomes hypervigilant after injury, it's less likely to get eaten. And again, you can put this out there purely theoretically, that perhaps back in the old days when we were getting hunted by lots of animals, if we were injured, we might be more likely to run away a bit quicker. But this is certainly not something that we would try in the lab nowadays. Okay, so hopefully I've convinced you that a bit about what pain is and why we actually do need it, even though it's unpleasant. What about the problem of chronic pain? So you might be sitting there thinking, well, I've talked about studying pain in rodents and in humans, and that all sounds rather dodgy from an ethical perspective. And firstly, to point out, everything to do with animal research in the UK, we're governed by the Home Office, the Animal Scientific Procedures Act in 1986, Amendment Regulations 2012. So nothing is done just for fun, but under very strict controls. But then I mentioned painkillers. People take ibuprofen. So why do we need to be researching pain? So, over to you. Is there a high prevalence of chronic pain in adults? Now, you can define chronic pain in different ways. The data I'm looking at are ones where we're defining chronic pain as pain in more than one body location that's lasted more than 3 months. So have a think. What do you think? Is it under 5%, 22%, 43%, or 64% for the adult population? So hands up for option A, under 5%. No optimists. 22%. 43, 64. So more pessimists, but you're right, it's 43. So this is a figure that's come out of a meta-analysis of lots of different studies. And this has been reproduced in different ways in different parts of the world, that by and large, 40-odd% of the adult population experiences chronic pain. Now, some of the younger members in the audience may be thinking that's absolutely ridiculous. But unfortunately, life tends to get worse. So what you're looking at here are data where it's been broken down by age. So for the undergraduate or postgraduate student in the room, the rate of chronic pain is quite low. As you get older, unfortunately, it does get worse. And that's not so much that pain itself gets worse, but rather conditions that can cause chronic pain. Age is quite often a risk factor. So is chronic pain highly prevalent? Yes. So I'm beginning to justify my existence as a pain scientist. We have a big problem. Just out of interest, if we look at what the cause of chronic pain could be, what do you think is the most common cause? So this is based in Europe. We've got 4 options: traumatic injury, herniated disc, that's the discs in the spinal cord, migraine, or arthritis, osteoarthritis. So hands up for traumatic injury. Herniated disc, migraine, arthritis, osteoarthritis, yes. By a long, long way. So these data are from 2006, and we've got a third of people experiencing osteoarthritis, arthritis as the most common cause of their chronic pain. This does not include rheumatoid arthritis, which is an autoimmune condition very separate from most other forms of arthritis, and this will definitely be, this bar here at the bottom will be a lot worse now because this study is almost 20 years ago, and the major risk factor for osteoarthritis is age. And obviously globally, as I'll show you later on, we have an aging population, so this is going to become, unfortunately, more prevalent, but from a pain scientist's point of view, great, even more people are suffering. So, obviously people suffering is bad, but we have painkillers, so surely, although we've got a big problem, we can just give them painkillers. So, over to you again. If we ask those chronic pain patients, is your pain medication adequate? Not is it perfect, is it okay? How many of them say, yeah, my pain medication is okay? Is it 91%, 72, 36 or 18? So hands up for 91%. Limited confidence in doctors. Okay, 72%. 36. 18. So I've lulled you into a false sense of pessimism, but it's still pretty poor. The important bit here is the blue bit. 2/3 of chronic pain patients say their medication isn't really doing them a lot of use. And the reason for that can be twofold. One, you'll give them pills, nothing changes. No one's going to take drugs for fun, or not for therapeutic purposes. And the other thing is the drug works for pain relief, but causes side effects. And if those side effects become unbearable, then you'd rather deal with the pain. You might know more about what you're dealing with. And just to give you an example, if we take opioids, which have numerous problems, but if we're talking about people who have bowel pain, so pain from their gastrointestinal tract, a major side effect of these drugs is constipation, which might seem humorous to some people, but if you've got a chronic pain condition being driven by your bowel not functioning properly, the last thing you want is a drug where one of the major side effects is to cause bowel problems. So, lots of chronic pain. Yes, we have drugs, but they don't always work. And then lastly, this idea that pain is only a sensation, that quite often you should just think about, well, it's ouch, just get on with things. And so data we published earlier in the year that was led by a postdoc, Luke, who's in the audience somewhere. You probably can't read this very well, so I'll walk you through some of this. What we asked in this big chronic pain survey of patients was how pain affected their everyday life. So, for example, down here at the bottom, in green means you agree with the statement, red means you don't agree. So my pain has a negative effect on my sleep quality. And virtually everyone with chronic pain says, yes, it definitely does. And we know that good quality sleep is important for good quality health. Poor sleep causes lots of other health problems. My pain status has a large influence over my motivation to leave home for any reason. Again, lots of people saying yes. So essentially, whatever we ask people about different aspects of their life, chronic pain was affecting their ability to engage properly, or how we would even a normal, happy life. And so pain is not just a sensation. So hopefully here I've justified my existence to you. Pain is common, the drugs don't always work, and has a major impact on everyday life. So if we can understand more about how pain works and how to potentially treat it, then hopefully we can get rid of this top and second problem here. So what exactly are we trying to do? Well, I'm going to walk you through 3 different areas of research going on in the lab to try and show you the sorts of things we can do and how we might be able to identify some new drug targets. So I've shown you this diagram before, the idea that pain starts in the periphery, but we can isolate cell bodies from the DRG and we can perform experiments on them as a model of what happens in the periphery. And so until now, it's been a bit of a background into pain. We're going to move a bit more in a scientific direction, so hopefully you'll be able to follow along, but don't worry if you don't, because there's going to be a summary as we go through the talk. So, if we're interested in pain from the joints in arthritic pain, we need to study those nerves that go to the joint. So again, we initiate pain in the periphery, signal goes to the spinal cord, it goes up to the brain and gets complicated. A very simplistic way to look at things is if we deal with pain out here, we can ignore the complicated bits. There's definitely an oversimplification, but that's what we're going to go with. Because it would be true, if you stopped pain at the periphery, the brain would never need to know about it. So we use mice as our model organism of choice, primarily because we can understand their behaviours very well and we can genetically manipulate them to look at specific genes of interest. Now, if we want to study nerves that go to the joint, one way of doing this is under anaesthesia to inject a fluorescent dye through the patellar tendon, so into the knee joint, and this will get taken up by nerves going to the knee and back to this dorsal root ganglion. So what does this look like? Well, if we take a slice through a dorsal root ganglion, here's the nerve fibres coming in, lots and lots of cells. If we now try and excite this fluorescent dye, we can see a few cells shining up. So very few cells within one of those DRG actually go to the knee. And if you think about it, that makes sense, because if you think about your experience of pain, if anything happens on your skin, you know exactly where that stimulus is coming from. And that's because the skin is the first organ to come into contact with potentially damaging things. You have many more nerve fibers there that enable you to detect what's going on. By contrast, you've got pain inside your body, so in your bowel or a muscle ache, it's much vaguer. You know roughly where the pain is, but not as precisely. And part of that is because you have far fewer nerve fibers there. And that's what you can see here. We have very few blue cells compared to the total number of cells that would be there. So if we're studying arthritis pain, we want to study nerves that go to the site of damage. So I mentioned osteoarthritis already earlier on. There's about 300 million people globally, and we have an ever-aging population. So we've got age on the y-axis here, and we're getting an ever larger population of people up here. And what you can see on the left is age against prevalence of osteoarthritis. The older you get, the more common it is. So it's estimated by 2050 we could have 1 billion people living with osteoarthritis. So how can we look at how osteoarthritis pain occurs? Well, I mentioned this sensitization process where the blue curve becomes the red curve. And we know a bit about different factors that can cause this. And one of particular interest is one called nerve growth factor, NGF. So this is known to be involved in making nerves grow during embryonic development, but is also associated with osteoarthritis pain. And lots of pharmaceutical companies thought this was a very exciting target. And so did lots of people involved in the pain world. But unfortunately, in 2021, the FDA and EMA said no. So we've got 39 trials across 16 years of this one drug called Tainzumab, and they rejected it. And the reason for this was not that it didn't cause pain relief, it definitely did. But a subpopulation of patients were developing rapidly progressive osteoarthritis. So it made their pain worse. They needed a knee replacement more rapidly. Now, there's one school of thought that says that's perfectly okay. If you have osteoarthritis and you know in 8 years' time you need a knee joint replacement, if you take this drug, you'll have a better quality of life, but you'll need your knee joint replacement earlier. Some people would be more than happy to undertake that risk. But the FDA and EMA said no. So at the moment, all this work that had been focused on this one particular substance we know causes pain has been thrown out the window. So that's either good or bad, because you can use that to say, okay, treating osteoarthritis can be difficult, we should all give up. Or you can use that as being inspiring to say, well, this failed, we need to find out more to identify what might be going on. Now, for any of you with cats and dogs in the audience, you're okay, because there's a company called Zoetis in the US which developed this type of drug specifically for dogs and cats, and they are treated quite happily with it. So cats and dogs have better treatment for osteoarthritis than we do as humans. Do the cats and dogs get this rapidly progressing osteoarthritis? We don't know. It could just be they won't live long enough for us to see it, but at the moment these drugs are clinically used and they seem to be doing perfectly okay. So, the better we understand pain mechanisms, the more hopefully we can do to develop new therapeutics. So if we want to understand pain mechanisms, we need to understand a bit more about how nerves are functioning. So here we've got an osteoarthritic joint. So we've got bones, we've got synovial tissue which has gone red, it's a bit inflamed. You get a fluid build-up quite often in the knee. A common way of causing or helping pain relief is to take out some of that synovial fluid to decrease the pressure within the joint. In green here we have nociceptors. They also change the way they innervate the joint during that disease process. And then lastly, you have these bumpy bits of bone. So the bone breaks down in osteoarthritis. You have a certain type of cell which tries to rebuild the bone, but it happens in an uncontrolled manner. And so these extra bits of bone usually aren't very helpful. If we zoom in into this middle part here, we can again see we've lost the cartilage, shown here in blue. That's disappearing. Got these bits of bone I was speaking about. And here's our nerve fiber being exposed to a different environment. In osteoarthritis, lots of different immune cells go into the joint. They release substances that change the activity of those nerves. And if we zoom in again, life gets really complicated. There's lots of different cell types releasing lots of different mediators, and that will trigger lots of different activity on the nerve to increase the sensation of pain. And so one thing we try to do in the lab is to understand what are the different processes involved, can we identify new targets? And one cell type I'm going to talk quite a bit about is what might be deemed a rather boring cell, the fibroblast. So most of these other cells here are exciting. When something changes, they infiltrate in, they change their activity, and they might try and attack a pathogen or degrade bone. Whereas the synovial fibroblast just sits there being part of the lining of the joint. under normal conditions. In osteoarthritis, in rheumatoid arthritis, these boring fibroblasts, these lining of the cavity cells, change their activity and can start to do more exciting things. And the reason that we are excited in them is because they are present in every single joint of every single person. So if we understand about their biology, they're perhaps more tractable for treating pain than some random immune cell type where one person might have a different immune response to the ongoing condition than another. So this synovial fluid contains all of these different substances. And so we first of all wanted to see, okay, well, can we study this synovial fluid in a bit more detail? Because not only do all these immune cells secrete things into the synovial fluid, so do the synovial fibroblasts. So patients with osteoarthritis have swollen knees, too much synovial fluid. They often have this removed as part of their therapeutic course of action and have steroids injected to decrease the inflammation. And quite often that synovial fluid is just ditched because it's not used to anyone. However, the patient can consent to having that synovial fluid analysed. And we were able to get hold of that synovial fluid. And when I say we, I primarily mean Sam Chakrabarty, a former PhD student, now postdoc in Germany. And what she did was she teamed up with Deepak Jadoo at the Rheumatology Research Unit. He was taking synovial fluid. Sam had a mouse model of the knee-innervating neurons of the mouse. And we took our culture of cells and we incubated them overnight with this synovial fluid. So the idea being we're trying to simulate the osteoarthritis joint. We've got our neurons going to the knee from the mouse, and on top of that we've left them sitting in the fluid coming from a human in pain. So how do you test pain in a nerve? Well, there are lots of different ways you can do it. We use a technique called electrophysiology. You put an electrode inside the cell and you can measure its response to different things. And the one simplest way you can measure how excitable that nerve is you can inject an amount of current and you measure at which point the nerve goes bang. And by bang, I mean this. This is what we call an action potential. You inject current in little steps and eventually it goes bang and gives you an action potential. That's the currency with which nerves send electrical signals. And so on the left, we've got a nerve under control conditions, and on the right, one that's been incubated with this synovial fluid. And so we're injecting small amounts of current in picoamps, but you can see a huge decrease in the amount of current we inject to switch that nerve on. So the human being with osteoarthritis is in pain. Nerves from a mouse that we incubate in the synovial fluid from those patients causes them to be more excitable. They're more easily switched on. But this isn't a particularly good experiment because we're comparing nerves sitting in control mediums, the sort of stuff they're always happy in, and then one sitting in this synovial fluid. What you really need to do is to compare to synovial fluid from healthy people. But most people would not happily volunteer their synovial fluid because you sort of need it for your joints to work properly. But you can obtain it from people who have given their body for medical research purposes, and that's how you can get out of healthy synovial fluid. And so if we look at these numbers here, we've gone from 550 down to 250. If we look at these three conditions, so this is just the averages here, we've got nerves sitting in normal medium, those in synovial fluid from people in pain with osteoarthritis, it decreases just like I showed you, and in the middle there are nerves taken from people who have no known joint disease. So synovial fluid normally doesn't cause pain, which makes sense because otherwise your knees would be in constant pain. But if we take it from people who are living with pain, it causes nerves to get switched on. And the question is, can we study that synovial fluid and its properties, how it changes between health and disease, to identify new mediators of pain? And one common mediator of pain in any inflammatory condition is acidosis. Now, this might seem like a weird thing, acid, but if you've been in the kitchen, you've got cut skin, vinegar or lemon juice gets into that cut, it stings. So acid causes pain. And in lots of inflammatory conditions, acid builds up at that site of inflammation. So what we're looking at here is pH 7.4 is the happy neutral pH. Osteoarthritis, rheumatoid arthritis, so we get a movement in the acidic direction. In acute lung injury, it gets far, far worse. But in lots of different conditions, acidosis occurs. And so back in my PhD, I was studying how does acid activate nerves. And unfortunately, as life has gone on, it's got more complicated. There are lots of different ways that nerves can sense acid. And so this is just giving you an overview. We have these things called acid-sensing ion channels, of which there are 6. That's what I did my PhD on. They were big things in the late 90s, early noughties. Then we have these things called TRP channels. So some of you may be familiar with one called TRPV1. David Julius won the Nobel Prize a few years ago for studying this ion channel. It's the one that makes chili taste hot. Chili tastes hot because it's got a chemical called capsaicin. Capsaicin activates this ion channel, which is actually one of the ion channels that is detecting heat. So chili tastes hot because it activates an ion channel involved in heat detection. There are some rather weird channels called K2Ps. These are usually helping to control the nerve in a sensible, non-excitable manner, but they get activated or get switched off by protons and can cause a nerve to get switched on. And then perhaps the least studied of all are these things over here, G-protein coupled receptors, which are activated by acid. So when you switch any of these on, you get a rapid change in the electrical activity of the cell. By contrast, in these receptors here, it's a slower change in the signaling processes going on inside the cell. And we had reason to want to study one of these proton-sensing GPCRs to understand its role in pain and how we can link that back to those non-neuronal cells I mentioned earlier on, the fibroblast-like synoviocytes within the knee joint. So we're going to focus on GPR65, one of these proton-sensing GPCRs. And this is all the work of a postdoc in the lab, Luke Patterson, who started this during his PhD and has just, as of Friday night, had this published in PNAS, so congratulations to him. So GPR65 is expressed in lots of non-neuronal cells, but it's expressed a little bit in nociceptors. Now the problem is if you put acid into an animal to see what happens, you activate all those different proton sensors I told you about. So what we were able to do is we were able to obtain a synthetic agonist which switched on this receptor, and we wanted to see if it could simulate how protons cause activation. So one of the things Luke did is he took all 6 of the proton-sensing GPCRs, and he used this drug, this BTB compound, to ask, does it generate a cell signaling response? And in orange, you can see, yes, in those GPR65 cells, not in the others. So we can use BTB as a surrogate for protons, because if you inject acid, protons, you'll activate so many different things. So now we've got a tool that enables us to activate one receptor that we know is present on cells within the joint, and this receptor is activated by acid, and in these conditions we have acidosis. So if you inject this substance into the knee joint of a mouse, you get inflammation. It's just a one-off injection. Over 7 days it decreases again. So we get inflammation of the knee joint. How do you measure pain in a mouse? Well, there are lots of different ways. One way you can do it is you can squeeze the knee joint of the mouse and you measure what gram of pressure causes the mouse to go ouch. So they withdraw their paw. You do similar tests in humans. If, for example, with a hot plate test, you ask them to say when it's hot, you move the paw. And you can see when we inject BTB, the force required to go ouch decreases. Now, the problem with this sort of measurement is people in pain don't go around poking themselves to see how much their pain changed from today to tomorrow. So we're also interested in studying how pain affects an animal's normal activity, just like we saw earlier on in that survey where we could see how pain affects humans' ability to engage with everyday life. One thing that mice like doing is digging. You put them into a new environment, they will dig and burrow and see what they can find. And we can see here that whereas normal mice just keep digging, those injected with this BTB spend less time digging. So BTB causes swelling of the knee joint, makes the mice go ouch at a lower pressure, and they become less engaged in activity they usually want to do. So the question is, how is this happening? So I've already shown you examples of how the ability to go ouch is based on how active your nerves are. If your nerves become more active, that will change your behavior. So if we put BTB onto nerves directly, and we're measuring different conditions here, but essentially nothing changes. The activity of those neurons stays stable. And that made sense because BTB is activating this GPR65, which isn't really present on nerves themselves. But I mentioned it's present on these synoviocytes. So what Luke did is he took, before I get to that bit, what we could show is in vivo, we want to see if the nerves change their activity. So these animals have been injected with fast blue to label their knee-innervating neurons like I showed you earlier on. Then we inject this inflammatory substance, we isolate just those nerves going to the knee, and you can see now they become more excitable. So we have more excitable nerves, and the animals also had pain behaviors. So in vivo, the nerves are excitable, but if we put BTB directly onto nerves, it does nothing. And that's where the synoviocytes come in. If we take synoviocytes from those animals, we apply BTB onto them, then we take the medium from those cells and put it onto the neurons. Now we can recapitulate that in vivo behavior. So what happens is those FLS are being excited to release mediators that activate the nerves and cause pain behaviors. And we were able to demonstrate using mice we've genetically modified to lack that receptor that everything was dependent upon that one receptor. But you may be sitting there going, well, this is in mice, what about in humans? So we teamed up with Simon Jones, who's at the University of Birmingham, who has access to human biopsy samples. So what we're doing here is we're using BTB to stimulate those FLS and see what substances they spit out. And essentially, compared to control conditions, if you stimulate with BTB, two cytokines, so chemical mediators between cells we know are important in pain, their levels get increased. So BTB can activate these cells to increase the sort of things they release that we know are involved in pain. And then the final thing Luke did was he went to take synovial fluid samples from patients with osteoarthritis, put it onto these cells, and we can see that some of these samples cause an increase in the cellular signaling. So this GPR65 can be activated by acid. It can cause pain when activated in mice. And if we take samples from humans who are in pain, we can use it to activate this GPR65. So we've got good evidence that this is a receptor that's involved in acid-mediated pain, it's involved in inflammatory pain, and we're now beginning to look at its role more specifically in more complex disease conditions. So I've mentioned earlier on the problem we have of using drugs. So this project would identify a new drug target, but is there something we can do that's a little bit different? So taking drugs orally, they go everywhere in the body, you have potential for systemic side effects. Can we target pain at the joint itself? So adeno-associated viruses can be engineered to target just sensory nerves. And we can also make them contain cargoes, enable us to control the nerve activity. So what we can do is you can inject one of these viruses into the knee joint and it infects the neuron. That neuron will now be expressing a new receptor, only in that part of the body because that's where we injected the virus. So when we then orally administer a drug, it will only activate the cargo that's carried by that virus. So we have a way of manipulating the genetic information of nerves at the site of injury and then giving a drug through the whole body, but it can only act at the site of injury. And so the idea is here can we have localized pain relief. And so this is a bit of a complicated system, so I'll try and walk you through it slowly. So this is teaming up with researchers at Oxford who made this virus, and we were trying to demonstrate how it could be used in joint pain. So we take our animals and we inject them with this virus, which takes about a month to label the nerves going to the knee. So we've got our 4-week period here, and then we give them an inflammatory injection into the knee. So we're simulating arthritis by causing inflammation. We measure their pain behaviors, and then we measure their pain behaviors again after we give them a drug, varenicline, which can be used in humans for treating nicotine addiction. So we're using a drug that's already been FDA approved, but this drug does something very special. It only activates something that the virus encodes, and that's a mutated receptor. So this is a receptor where people have taken 2 different ion channels and mixed them together. And the important thing is they've mutated part of the ion channel that recognizes a natural substance in ways that it no longer does. It's only activated by this synthetic compound. So essentially, you use the virus, you infect the knee neurons, they make this new ion channel, and nothing happens until you inject a drug to activate it. And this is just showing in blue here, we get these knee-innervating neurons being labeled. So we've got 2 sets of animals. We've got animals that have given a control virus, because we need to make sure the viruses themselves don't do anything, and then viruses that have got this thing called a PSAM. This is this thing we're going to activate with varenicline. We inject this inflammatory substance just into one knee, and we get inflammation in both the control animals and in those with our special virus. We put these animals on a rotor rod just to see how well they run. We want to make sure the knee injury doesn't cause them problems. We want to make sure the virus doesn't cause them problems. And they all run equally as well as each other. So these animals are not crippled, it's a mild joint inflammation. What does change, though, are their behaviors. So what we're doing here is we're squeezing that knee joint, and the important set of data are here. Under control conditions, they go out at 300 grams. After inflammation, it goes down to 150. On the right, when we give this activator of this special virus construct, it goes back up. So we can relieve the pain. The pressure required to make them go ouch changes. I also mentioned how we like to study the digging behavior of mice. We see the same thing. Again, if you focus on the data on the right, Animals will dig for about 20 seconds across 3 minutes. After inflammation, they dig an awful lot less. We activate this receptor, it recovers. So we can change an animal's behavior by activating this receptor that's only present in knee-innervating nerves. And importantly, we can see that it's an activation property of the nerves that causes the behavior, because we measure the function of those nerves. If we apply this drug, it makes the Rio base increase. It's the same measurement I've shown you before. If it goes down, the nerve gets more excitable. If it goes up, it gets less excitable. And so that's what we're seeing in this situation here. Okay, so a tale of painless delivery. What exactly does that mean? I've shown you this picture before, and I've shown you that genetics can cause you to have no pain sensation at all, and the problems associated with that. And one of the causes is mutations in this gene, SCN9A. This is a very strange gene because other mutations can make you have lots of pain. The pictures here where you can see is very red and swollen peripheral parts. This is familial primary erythromelalgia. So just like this is a congenital condition, so is this condition here. And these people have a burning redness, burning pain and redness of the extremities. So we know that genes can control pain, and these are very extreme conditions. We wanted to try and study a population of people to identify other genes that could regulate pain. And so this is a big team effort, lots of clinicians, lots of scientists, and we were asking, can we find genetic variants that control pain through studying painless labour? Now, obviously, I've never given birth, so I don't know, but from what I've seen, giving birth seems to be painful. But it's well known in the clinical world that some women say, no, it doesn't hurt. When you rule out psychological, cultural, whatever reasons, there are some people who say, no, it just didn't hurt. So we studied across maternity wards women who said, no, it didn't hurt, they didn't ask for analgesics, but were otherwise healthy. And we then said, okay, that's great. Can we please have some DNA from you, and can you please come into the lab so we can hurt you? And some of them said, yes. So when I say hurt you, this is technically called quantitative sensory testing. So you're exposing people to different stimuli, and you're measuring when do they go, ouch, after they said that is okay. So on the top row here, we're looking at your ability to detect cool, warm, and pressure. So not ouch, this is just your thresholds for normal stimuli. And at the bottom, we're looking at cold ouch, heat ouch, pressure ouch. So in white, the see-through circles, these are our healthy, controlled, normal people, as it were. And in black, we've got people who had not had any pain during labour. And you can see their threshold for going ouch to cold is lower. It needs to be a colder temperature. For heat, it needs to be hotter. And for pressure, it needs to be higher. These aren't huge changes. These are not people with no pain, but it's a subtle change in their pain sensitivity. When the women were questioned, most of them didn't think they'd had any unusual pain phenotype. A couple said they'd been known in the family to have a high pain threshold, but it's subtle. It's a small change. So what exactly is going on with these women? Well, most of them are in black circles, but there are 3 red triangles. And I briefly want to say what these 3 red triangle women represent, because they all had the same mutation in one gene. And that gene is something called KCNG4, which is an ion channel. So we've come across this term before, but essentially these are molecules that sit in the membrane of nerves, and they control how electrical signals are sent. They function a bit like a lock gate in a canal, but they let ions rather than water through. So when they open, the nerve gets switched on or switched off, depending on the ion channel, and different variants can control how much they open, how often they open. And as I've already mentioned, local anesthetics like lidocaine, they switch off one set of ion channels. That prevents nerves firing. So when you find a genetic variation in humans, that's great. It's an observation. You've got women, in this case, with a phenotype and a change in their DNA. But you can't prove how that change in DNA causes that particular phenotype. And that's where the mouse mechanistic work can come in, because we can take nerves from mice and we can change which forms of this ion channel they produce. The normal form or the rare variance form associated with less pain. And that's exactly what we did. So again, we're injecting current to switch the nerves on. We've got the normal version of this ion channel at the top, the rare variant at the bottom, and you can see we have to go higher up this ramp to cause the nerve to fire. And that's what's summed over here. This is the normal version, this is this rare variant. We need a much higher input. So essentially, these women, their nerves work. They just need a higher input to make them go bang, to send that signal, which correlates exactly with what we observed on the pain tests. They had a heat pain threshold, it was a bit higher. And so we're thinking we can use this KCNG4 as a drug target because these women were perfectly healthy. So you can go from human genetic data to say, okay, this gene is involved in pain, the women are healthy. So if we target this gene in other people, we're not so worried about side effects. So it's not if we're going to trigger epilepsy or something like this, because we know these women are otherwise healthy. All right, in the last 3 minutes, we're going to talk about naked insights. And that's because I work with these animals, naked mole rats, and I feel I can't possibly give a talk without mentioning naked mole rats. So bear with me if you will. These are subterranean rodents that live in Ethiopia, Kenya, and Somalia. They're eusocial. So they have one queen, potentially 1 or 2 breeding males, everyone else is a subordinate. They're cold-blooded mammals, so I have to keep them in a room at 32 degrees to mimic the environments they've evolved in. They live for over 35 years. The record holder is 39. They show no age-related increase in death risk. They also don't show many signs of aging. This is me, aged 38 at the time. I've got grey hair and wrinkles, and I'm holding a 37-year-old naked mole rat. I can tell he's an old mole rat, but most people wouldn't tell the difference between a 1-year-old, and a 35-year-old. And this no increased risk of death is shown here. We've got how big you are against how long you live. The bigger you are, the longer you should live. Elephants live longer than mice. Mice live about 2 years. Heterocephalus glaber, different-headed bald thing, the naked mole rat, they live for 35 plus years. So why are we interested in these in pain? Well, several years ago, we could show that they had normal responses to heat. So we're looking at an animal going, ouch, in a mouse or a mole rat, same response. Capsaicin, that substance in chili peppers that tastes hot, they don't care less, and they don't respond to acid. And I've already shown you how acid can cause pain. We saw how that blue curve shifted to the red curve. In naked mole rats, you can give different substances that cause thresholds to change in mice. Mole rats don't care. So that's great. You've got some odd-looking animal where you've got this strange behavior. Well, if we study that behavior, we can understand the molecular basis of it. So we're able to identify one gene that controlled this acid insensitivity to pain, one gene that can control this change in sensitivity. By studying extreme biology, we hope to understand more about normal biology. Mole rats are resistant to osteoarthritis. We have a new project in the lab trying to understand how is that possible. We've recently shown that their gastrointestinal tract shows very tight permeability. So lots of gastrointestinal diseases, such as inflammatory bowel disease, have a leaky gut, not so the naked mole rat. So hopefully I've convinced you that nociception pain is unpleasant, but overall it is good for you. Sometimes, though, it goes wrong. Chronic pain is common and poorly treated. And studying how pain works is going to be critical in developing new therapeutics. I should put my hand up to say I'm obviously in a pharmacology department, so I think drugs are great, but there are other things you can and should do for managing your pain. We try to integrate human samples and the genetic base of pain in humans, because obviously if you can find something in humans, the chance of translation from bench to bedside are much higher if you're only studying cells, or in our case, mainly mice and mole rats. And indeed, studying extremophile animals could show us lots about pain and beyond. So just to acknowledge that I spend most of my life answering emails and filling in forms. These are people who actually do all the work, thanks to lots of people giving us money over the years and a wonderful group of collaborators, many of whom are clinicians with whom we wouldn't have access to our clinical data. Thank you very much.